My name is Sheila Guillotone, and this is the Simsbury Woman. Today we're going to explore the world of science and technology, specifically the science of genomic medicine. For many of us, myself included, knowledge of the genome comes from watching crime dramas on TV, where discoveries about DNA almost always solve the crime. But that's not the real world, and I'm delighted today to have as my guest a real scientist who will try and give us a more accurate introduction to the science of genomic medicine. Please welcome Yu-Hui Rogers, Site Director of the Jackson Laboratory for Genomic Medicine, located in Farmington, Connecticut. She's a Simsbury resident, and I'm so happy to have her here as my guest today. Yu-Hui, please tell us a little bit about your background and the history of how you came to the U.S. and your educational background.
Thank you, Sheila. So I came from Taiwan, and I came to this country about 25 years ago after I finished my college education. And as an Asian female scientist, I somehow feel a little bit suffocated, from where I came from, because I came from a very traditional family. And coming to the U.S., I must say, for graduate education, I must say that is a liberating experience, and I have not regretted for a moment. So I came to the U.S., like I say, about 25 years ago by myself with a suitcase, not even really knowing how to speak the language. So I spent the first year when I came here in English Institute, learning English. And then after that, I went to graduate school. And I'm a chemist by training, an organic chemist by training. And after graduate school, I would say that I stumbled into the world of technology development for genetic testing, and then move on to working on genomics science. So I spent about the past 20 to 25 years working as a scientist before I came to Connecticut. And I have been involved in some really quite exciting and some might consider groundbreaking projects.
Really?
I was part of the team that actually sequenced the first human genome back in 2001. And I was also involved in some other really quite exciting projects, including a global ocean sampling project that we actually sail around the world collecting seawater samples and trying to understand what organisms are in our great ocean.
Okay. That all sounds very complicated, and we're now almost evolving into environmental issues. So before we go there, could you kind of explain a little bit about the difference between genetics and genomics? Most of us, I think, have a concept of what genetics is.
Yes.
But how does that relate to genomics, and what's the difference?
Right. So maybe we can actually go, from the more basic, starting from, our cells. So within our cells, basically their nucleus, the center of it, they are our genome. Basically, every single living thing in the world has a genome. We call it the code of life. Basically, it's an instruction book that actually direct the building of the organism as well as the function of the organism. So a genome, there's actually a graph that we can actually share. So this is the cell, and inside the cell there are our genomes, and the genomes are organized in chromosomes. So I think that many of you actually, most of you probably know that we each have 23 pairs of chromosomes.
Right.
So it's almost like, if your whole genome is a book, chromosomes are the 23 chapters of the books.
Okay.
And within those chapters, there are sentences, and those are the genes. So specific genes actually encode specific proteins and specific functions. Okay. And then you go down actually deeper, basically, it would be individual bases that we call nucleotides. Basically, our entire genome is 3 billion base pair, and it's all made up of 4 different bases, A, C, G, T.
Okay.
And that entire chemical structure is the DNA that we often refer to.
Okay.
Did that give you a reasonable sense in terms of...
When I look at the picture, it definitely helps.
Right. So the genes are almost like sentences.
Okay.
And the bases are almost like the letters.
Okay.
So, you have your...
so you're breaking it down into smaller and smaller pieces.
Yes. And the whole thing is a genome that is the instruction book or the recipe book.
For being human?
For life. For all lives.
Okay.
Yeah. So so like I say, every single living thing has a genome, including plants. Yes.
Okay.
Yes.
All right. Okay. Now you were starting to talk a little bit about some of the projects that you were involved in. One of them kind of links back to what we were talking about, the DNA on crime shows.
Right.
You were involved, we'll get to the sequencing of the genome later, but tell us about that particular research project that you did that relates to my crime show analogy.
Right. So I started out my career actually developing technology for genetic testing. And interestingly, as you mentioned, I actually work on a specific type of genetic testing. It's actually for paternity testing. And that actually goes back to your original question regarding, what's the difference between genetics and genomics. So with genetics, basically, we tend to look at basically, going back to the analogy with the books, we look at specific sentences. We don't read the whole book.
Okay.
With genetics, we actually going to look at specific genes or landmarks that we call, within the genome, and trying to use the information to make some biological interpretation of, what it meant. So, like I say, my first job, we actually were developing a panel of genes that we can actually use to determine paternity between individuals.
Okay, and this is, if I believe the television shows, this is actually quite accurate now.
Oh, yes, very, very accurate. Basically, if you have enough numbers of, we call it loci, basically, number of sites that you actually look at, basically, it gives you some sort of power of exclusion.
Okay.
Basically. Yes, it is really quite accurate.
Okay.
And another association that I have with more of, forensics type of DNA technology is related to my previous job. After we sequenced the human genome in 2001, and as many of you remember, the World Trade Center event actually happened.
The World Trade Center, right.
Right, 9/11. And my company actually committed to developing a forensic sequencing pipeline where we actually look at the mitochondria DNA. Mitochondria DNA is actually particularly good for specific type of forensics testing. So we actually look at the mitochondrial DNA and we develop a high throughput sequencing pipeline with basically in attempt to actually help identify the World Trade Center victims.
So this was identifying the victims even when you just had dust, basically?
Yes, and that's why we look at the mitochondrial DNA. Mitochondrial DNA is small and it's circular. So our DNAs actually do break down very easily, especially, under extreme environment, high heat and so forth. But mitochondrial DNA has ability to actually stay more intact because it's small, it's circular. And it's actually often being used in the forensic setting, for difficult to identify individuals.
Okay.
Yes, and we can actually, so in order to actually identify the individuals, we also need to be able to sequence and get sequencing information on the possible family members.
Okay.
So we can actually match the information.
Okay, so you would get samples from family members, and then in the lab,
yes,
you would be able to identify
we can determine the kinship. between the individuals. Yes.
Wow, that's impressive. You also talked a little bit about another research project that you did involving the oceans. And
yes.
again, we're going back to the genome and every living thing having a genome. Tell us what you did. You said you traveled around the world?
Yeah, so I was actually part of the team, basically that we have our boss had a large yacht, and he actually converted it into
that's nice.
into a research vessel.
Okay.
And we actually took the yacht around the world, and every 200 kilometers we stop and we actually collect the seawater sample, because there are a lot of organisms inside the ocean.
Okay.
Microorganisms, basically bacteria.
All of which have
fungi.
Genomes.
Yes.
Okay.
So we
I learned that much already.
We actually filter the seawater through filters.
Okay.
And capture the organisms on the filter, and we bring the filters back to the lab, and we actually extract the DNA of the organisms from the samples that we collected, and then we sequence the whole thing.
And then what is that going to tell us in the end?
So in the end, you are able to actually identify with more abundant organism. If you have enough information, you can actually get the whole genome assembled.
Oh.
And some of the organisms that are not abundant enough, you will at minimum actually get genes. Okay. Basically, interesting genes. Basically, a lot of them have very interesting biological functions. Mm-hmm. For example, there are some organisms in the ocean, they can take the sunlight and process it and produce hydrogen. Mm-hmm. So one can imagine that we can utilize information like that for bioenergy production, for example. Oh. Yes.
Okay.
Yes. Or actually, there are also other applications, for example, bioremediation.
Okay,
so,
you're going to have to define that.
Right. So for example, oil spill.
Okay.
And so you have the oil spill, around the coastline or in the deep ocean, mm-hmm. and because the oil actually changed the ecology, the environment. Mm-hmm. Many organisms actually die, or the ecology gets damaged. Mm-hmm. However, there are certain organisms that have the ability to actually go in and clean up the waste.
Really?
And allow the ecosystem to be renewed.
Okay.
Yes.
Okay, so that's how when we hear years after a tragedy and people go back, it's like, wow, there was really, some environmental disaster here, but things are growing again and the water's clear again.
Yes. Okay. Yeah. And if we really understand how these bioremediation processes work, we can potentially speed up the recovery.
Oh.
Yes.
Okay.
Yes.
All right. Okay. We sort of went off on a different track. One of the things we want to talk about with you specifically is the research into genomic medicine, which I believe the point is to develop drugs or treatments that will actually cure or treat diseases.
Right.
Okay. Now, you've been involved in cancer research, I think.
Right. Actually, me personally, I do not, I'm not directly involved in the research.
Okay.
Actually, my role is more of an administrative and operational. As a site director, I provide support to the scientists that actually conduct the actual research. But I can actually briefly talk about a very high level in terms of the type of research that we do related to genomic medicine. So cancer is definitely a key area. We also look at cancer immunology. We also look at human microbiome. We also look at diseases such as diabetes and heart diseases, basically just to name a few. But specifically to cancer, I think that you and I have talked about basically, cancer is almost like weeds, growing in your, lung, basically. And sometimes, they get out of control.
And that would be the cancer cells.
Yes,
Okay.
they continue to grow and expand, and by the time you know it, they take over your lung. And what we do is really understand, what kind of cancer cells are there, and in terms of the biological mechanism and pathway, how they developed, and potentially how to interrupt the pathway so that we can actually slow down the growth or basically completely eliminate the cancer cells. And a lot of those information is actually genomic information. And I can probably use an example that most people are familiar with. Basically, most of our audience probably have heard of the BRCA1 gene or BRCA genes.
Right. Angelina Jolie.
Yes,
Got
yes.
it.
So
Okay.
the BRCA genes are actually linked to breast cancer.
Okay.
And BRCA gene is basically, there's actually BRCA1 and BRCA2. So they actually are cancer protein suppressor genes. And some individuals, their genes have we call mutations. So almost like basically you have a misspelling in the sentence.
Okay.
And therefore, it doesn't function properly. And that will actually increase the susceptibility of the individual to potentially get breast cancer and other cancers too. Basically, in fact, BRCA1 and 2 have been implicated for other cancers as well.
Oh, really? I thought it was just breast cancer.
Right. So, but the actual genetics is actually really quite complex, and that's why a lot of times, when individuals actually take genetic tests, it's really important that they actually work with the physicians as well as the genetic counselors to actually understand the results.
Okay.
And what does it mean, to them?
Now, so your research then leads to treatments and potentially even drugs?
Yeah, so certainly identifying the pathway, the target, for example, and the pathway that we can target to actually develop treatment.
Okay.
Yes.
Okay. You told me a story, which I would like you to share with our viewers, about the importance when you're developing drugs and targeting. And of course,
Right.
your genomics is what's giving you the roadmap
Right.
for the development of these new drugs. But tell our viewers what you told me about the development of a specific cancer drug that was developed by two labs, very similar, but what happened in the end?
Right. So just actually very recently, Merck is a large pharmaceutical company, and many of you are aware of. Basically, I believe it was just early summer, they had a cancer immunotherapy drug that was approved by the FDA. They passed a clinical trial. And it's really interesting. I was actually reading a news article from Bloomberg, and they were actually citing that result and basically really discussing basically their approach in terms of how they went about the clinical trial. Merck actually went with a targeted approach. What it means is basically that specific treatment is actually targeting a very specific protein.
So is that a specific kind of cancer or a specific type of individual?
Lung cancer.
Oh, okay. So
Lung
it was
cancer
a specific
and-
type of cancer.
Yes, and a specific population that has that cancer actually have that protein, I believe, at elevated level.
Oh.
So they actually went about the clinical trial basically targeting a specific group of patients that had elevated level of that protein expressed. Basically, I think the cutoff is at 50% or something like that.
Okay.
But in order to actually diagnose and stratify the patients, they also need to do a companion test, obviously, basically, really to detect the presence and also quantify the protein. So that's the way they went about doing it. And the news article actually was, basically, discussing, their approach and basically say that, well, yes, they went with a more targeted approach, but that also narrow their market. Okay. So that it's going to take them, a little while to actually get up to, a certain amount of revenue. And on the other hand, there's another pharmaceutical company, BMS, BristolMyers, actually has a very similar drug.
Okay.
But BMS actually went with a different clinical trial approach. They target for a broader population. So instead of testing the presence of that protein at a certain level, they went with a much broader population. And with the hope that they can actually market and sell the drug to actually treat larger population, and
hence more profit.
More profit, faster. That's the implication. Right. But I believe in August, BMS clinical trial failed.
Really?
Yes, and I think that really, that kind of speak to basically, it's a great example for actually precision medicine that some of you might have heard. Basically, certainly, precision medicine is not just about genomic medicine, but genomic medicine is definitely a big driver for it. So a lot of things that we do within our laboratory is really, really understanding, the genomic basis of these diseases and knowing what pathway to target and so forth.
So it sounds like the more targeted the research or the trial, the more likely it is to be able to eradicate or treat the disease. But that's going to make every disease you're going to have to basically do that for every disease or every class of diseases?
So it depends on what it is.
Okay.
Certain things are more, more widely applicable. Certain things are more specific.
Okay.
For, depends on the diseases.
Okay.
Depends on the diseases. Yes.
Okay.
Yes.
So what do you see in the future? I mean, are we actually using genomic medicine? Are we going to see the ability to cure most diseases? Down the road a bit?
I think that we're going to be able to start understanding a lot better.
Okay.
And we are going to be able to come up with more innovative and interesting discoveries. Our genome is actually really dynamic.
Okay.
It's
what does that mean?
Right. So even just looking at the cancer cells themselves, right? Basically, many of you probably heard of resistance, right? Drug resistance, basically.
Okay. That's a big thing these days with antibiotic resistant.
Yes.
Those are genes.
When you actually develop antibiotics or some sort of vaccine or different type of treatments for different infectious diseases, and a lot of those microorganisms and viruses, they evolve really fast and they develop resistance.
Okay.
And cancer, on the other hand, I don't necessarily want to get into too much detail,
right.
But similar to the wheat and the lawn analogy, basically they are cancer stem cells, basically.
What is a stem cell?
So stem cell actually has the ability to actually proliferate and evolve into different cell types.
Okay.
Basically, and then also cancer stem cells, basically, you could potentially actually treat the cancer, but if you actually miss the cancer stem cells, later on they may actually proliferate.
Oh.
And they may actually not be in abundance, in the early stage. So it is a really, I would say that our genome is really dynamic, and yeah. So
we're not quite at the point where you guys are going to be able to simply say you have this kind of cancer and we can give you this specific drug to cure it.
We're working towards it.
Right.
But I would say that this is really only the beginning.
Okay.
Of us actually really starting to understand our genomic makeups, and we only are beginning to have some ability to collect a lot of genomic information. It's far more complex than we anticipated. We spoke about a little earlier the first human genome that we sequenced back in 2001 actually cost a lot of money.
I'll bet.
So,
but didn't people sort of think at that point that, oh, now we're going to understand everything?
Yes, we were a little naive.
I see.
So,
Okay.
It's hundreds of millions of dollars that when we actually when we sequenced the first human genome, basically, there were two versions that were sequenced. One is the public version, another one is the version that was involved with Celera Genomics. Basically, just the sequencing cost itself is $100 million. So that does not include other overheads and analysis.
That's just reading the book.
Yeah, it's just reading the book.
Okay.
$100 million.
Okay.
But now...
and now you have to do something with the book.
Right. And we need to read a lot more books to really understand our genome to the degree that we can really have a better handle on a lot of those molecular mechanisms that actually impact diseases, yes.
Are there some diseases that you think are more likely to respond, or is it just the more popular ones are being addressed because there's more people who have them?
I probably wouldn't be the one to answer questions like that, but I definitely see that a lot of times scientists actually respond to basically more of the policy and funding environment too, right? Basically, if there are specific diseases that's really impacting the economics as well as the finances of the country as well as the state, I can see that policy tend to actually be made so that there's encouragement to focus on diseases that have much larger social costs.
Right. Okay. But unfortunately, even within that group, say cancer, I mean, you've got, as you said, so many different kinds. It's not something that's just you're going to find a cure for cancer and it's going to cure all of them. They all have to be addressed individually.
More likely you're going to have to address them based on the type and potentially even with population, basically as we discussed. So, and that's why it's becoming more focused and becoming more precise in terms of and that also means that it becomes more effective.
Okay.
Right.
Okay. Well, unfortunately, we're getting close to our time being up. I hope everybody has enjoyed this very brief look into science, and especially the science of genomic medicine and what it can do in the future. There's so many applications that we didn't even think about, including environmental. I especially want to thank my guest today, Yu-Hui, for giving us her time and sharing her extraordinary experiences on learning and watching the advancement of the health benefits and the research done by facilities like the Jackson Laboratories. I wish every one of our viewers a very happy and a healthy day. I thank you again for coming, and thank you all.
Thank you.
Funding for Simsbury Community Television is provided in part by contributions from viewers like you. Thank you.